Thank you very much for that very warm welcome. I'm delighted to be here to celebrate the contribution of Dr. Borlaug, but I'm also very excited about joining the CGIAR at also a very important time, but also a critical time, and I look forward to working with you in the future. It's been really excellent to be here over the last week and to see the excellent work that's been done in terms of the wheat program, but also speaking to some of your colleagues working on maize breeding. So I've not formally started my new job, and although I was given a deck of slides, I'm going to completely ignore them, and I'm going to try and answer this essay question by focusing on three interrelated areas, some of which have been touched on already. The one is the context in which we're working currently. Secondly, the scientific landscape. And third, the political and social agenda. And all of these are important in attempting to answer the essay question that I've been set. We've already discussed this in detail, but this is a slide from David King, the former chief scientist of the UK, that really outlines the 21st century challenges. And I think there's two or three key things from this cartoon that we need to note. One is that these issues, these problems are interrelated. As we heard this morning, food production is not the same as food security. The second thing that we really need to, I think, appreciate and understand is that scientific innovation is going to be critical in addressing these big societal challenges. And finally, in order to address these big societal challenges, partnerships and collaboration are going to be absolutely critical to go forward. In terms of the scientific landscape, I think we're living in a period where there's an explosion in our scientific understanding of the life sciences, but also in terms of the physical sciences as well. And the interaction between the life and physical sciences are giving us new opportunities to tackle the big challenges we see, but also to combine excellence in terms of that research with the translation of it to deliver impact. And I think this is a wonderful opportunity for the CGIAR, and I also think that it has really, in many ways, rejuvenated and rekindled an interest in agriculture. Of course, one of the principles that was discussed yesterday is sustainable intensification, which was highlighted in a report from the Royal Society in 2009. The other aspect of this, though, which I think is really important, is that institutional capacity and capability coupled with agility is going to be really important. The third aspect that we need to consider, of course, is the biology of the species we're working with. And in the case of wheat, we're dealing with an inbreeder, a founder crop of civilization, but also an alloexaploid represented by diploidization and tetraloidization of many diploid species, providing buffering capacity, which again provides opportunities. But it means from a breeding and a crop improvement perspective, we need to be thinking about these things differently, depending on this breeding system and depending, certainly, in terms of delivering for sustainable intensification. Now, Dr. Borlaug, in addition to delivering new varieties and really creating the Green Revolution, essentially what he did was really establish the CGIAR as a major center for innovation. And what in common language today was a transformation step in terms of turning wheat into a major crop that fueled the Green Revolution. Now, alongside the evolution of what I've just been describing, there's been an evolution of the CGIAR. And many of you in this audience are better equipped to comment on this than I, except to point out that in the 1960s we were dealing with 3 major commodity institutes, and now there are 15 delivering across a whole range of activities with an attempt to bring a systems-based approach to the delivery of major outcomes, particularly around 4 system-level objectives. So during this period of the 1960s to present day, there's been many institutional changes, and those changes are still taking place at the moment. One of the other factors that is important is that there's been an increase in funding, and now approaching $1 billion in terms of investment in the CGIAR, which I think again requires a responsibility to be able to use that funding wisely and efficiently and also in a way that is going to be creative to address these big challenges going forward. Now, the way this research is delivered at the moment are through 16 research programs, of which wheat is one. And as you can see from this slide, there are many major goals and objectives encompassed within the wheat CRP. And having spent much of this week with the wheat CRP scientists and researchers and plant breeders, I think the wheat CRP is very well positioned to address some of these global challenges. What I want to do now actually is to step back from it and talk about a little bit about some of the activities I've been involved with when I was at the National Institute of Agricultural Botany, and then come back to these questions towards the end of the presentation. The first thing that we did some years ago actually was pose the question, how is wheat performing relative to maize? And in this slide here, if we think of the x-axis as the wheat yield, and then the red line plotting the performance of maize against wheat. And what you can see is from about 1998 onwards, a massive divergence between the performance of wheat and the performance of maize. Now, I'm sure, as we heard this morning, there'll be many reasons that can explain this, or can certainly put forward some ideas. But one of the things that we started to do at NIAB was think about genetic diversity and actually learn from some of the activities that were going on in CIMMYT. So in about 2005, Andy Greenland and I came here to look at a synthetic program in CIMMYT, and then went back to Cambridge and established a synthetic program there. However, before doing that, we carried out a fairly detailed genetic analysis of potential D-genome donors to identify the spectrum of variation. And as we pointed earlier in a paper in Genetics a few years ago, that there's at least 2 clades for the D-genome donors. But also the opportunity to sample a broader spectrum of variation, which was complementary to the synthetic material that had been created at CIMMYT. We coupled this by looking at diversity in the tetraploid donors, and looking at not only nuclear diversity, but cytoplasmic diversity. And my colleagues now at NIAB have been able to go on to create synthetic wheats, which are really performing very well under both high intensive conditions and low nitrogen conditions, as is shown in this slide here. So that's a little example of something that we learned from CIMMYT and something that we applied to the program in the UK. The second point that I want to talk about briefly actually are the RHT dwarfing genes and the long connection that's existed between CIMMYT and the UK. In this slide here, I'm summarizing some very influential individuals in terms of the production of the identification of the RHT dwarfing genes, including Mike Gale and also Nick Hubbard, who actually cloned the A genome homolog of RHT1, which is codes for DELA protein, which actually is a transcription factor with a premature stop codon, which actually results in GA insensitivity and reduction in height. One of my students, Ed Willem, actually cloned the 2 other homologs of the RHT dwarfing genes. And this is summarized in this cartoon here, where he looked at the diversity in the A, the B, and D homologs across several wheat cultivars. In this cartoon here, we can identify the deletions shown in triangles and some of the stop codons shown by the red stars. The important point that I want to make from this is that this allowed the creation of a haplotype dictionary. And I think if Norman Borlaug would be alive today, I think he'd want to be using this sort of information to start not only interrogating germplasm, but also to be able to look for those key recombination events, which he called earlier belly-up chromosomes, I think was the term that was used. But really what I want to say is that this is a time when we can tackle problems that were really difficult to tackle previously. The other thing that Ed Willem was able to do was actually try to track the ancestry of the Norin10 dwarfing genes. And this was a kind of an interesting study and a bit of a puzzle, because we were not able to trace back the individual RHT dwarfing genes from Norin10 to its putative progenitors. Now, there may be many reasons for this. One would be, of course, the heterozygosity of the genetic stocks, the fact that the pedigrees may have been wrong. But I use this to illustrate the fact that really knowing germplasm is critical, but also being able to interrogate that germplasm and certainly to sequence and resequence genetic material is going to be really important in the future. Now, as we know, wheat has got a very, very large genome, and the fact that we're dealing with 3 genomes, there is heterozygosity or differences between the 3 genomes. We have polyploidy, also large repetitive regions, but just as we saw in rice, actually sequencing the rice genome, having a reference genome to resequence is transforming the way not only breeding is done in rice, but the way in which biology is done in rice. And I think we're entering a phase where the same opportunities will be available for wheat. In this case here, this is a slide from Genome Analysis Center in Norwich, which identifies the progress on sequencing of the chromosomal segments of wheat. And there's now a period where that sequence information is going to become much, much more enabling and will give opportunities for wheat breeders that we've never had before. And this is a massive opportunity for the wheat CRP to really drive the agenda in using that genome knowledge to really produce not only new products, but also the knowledge that's going to be required to deliver on the big societal challenges. Indeed, I think if we are creative about this, breeding programs can be used not only for the generation of new products, but they can be used for testing hypotheses and can be a real magnet for attracting talent into the wheat genetics and wheat breeding community. What is also important, I think, if we are going to address this issue, is to start thinking about population design. And this is a summary slide of some work that one of my colleagues, or former colleagues, Ian Mackay, did in terms of developing magic populations, multi-parent advanced generation intercrosses. The significance of magic, which is very similar to NAMS with 1 or 2 subtle differences, is that it gives you an opportunity not only to increase resolution, genetic resolution, but an opportunity to look at the distribution of haplotypes across recombinogenic regions of the genome. And this is providing an opportunity not only to isolate genes controlling complex traits, but also to be able to develop populations such as that are necessary for genomic selection to deliver genome-based selection in wheat breeding. What's also important about this actually is the crossing design that you use. And Mac developed a system which he really stole from incomplete designs and Latin square classifications to really think carefully about the design of these crossing programs so you can trace individual ancestry and individual pedigree back to one individual. And that's a very nice system which I think will complement the utilization of genomics in wheat breeding, but also to be able to enable the isolation of genes controlling complex traits. And I think you heard earlier in the week a talk from Alison Bentley, certainly using this population to isolate and clone genes responsible for adult plant resistance in yellow rust. So let me finish with 2 slides on the CGIAR research programs. Apologies, these are fairly high level, so apologies for that. But I think if we look at CGIAR research programs and look to the future going forward, I think we've got to be thinking strategically about this in terms of identifying ambitious programs that are tackling problems that are beyond the reach of others. We've got to embrace this issue of sustainable agricultural intensification, but providing as well a framework around products, public goods, and knowledge. I think institutional capacity and capability to deliver is going to be really, really important with a strong partnership ethos. And again, the leadership and management commitment to manage the complexity, not only within breeding programs, but also the complexity of the range of partners that are going to be needed. And I think finally, this need to grasp the issue that agriculture, modern breeding is data intensive, and managing that data in a way that's going to deliver the outcomes we need is going to require investment, not only in terms of capability, but in terms of skills as well. I think, again, if I drill down on the wheat CRP, what a wonderful opportunity to provide intellectual leadership for 21st century breeding. I think to do that, we've got to have an interface between the statistics, informatics, and population genetics in order to take these things forward. Got to be maximizing the opportunity for the exploitation of genetic resources in breeding and a proactive management of diversity in breeding. Why? Well, actually, wheat is very different to maize, that's obvious, but we've heard a lot of emphasis in terms of introducing variation to breeding programs. In wheat, that's not the challenge. It's actually managing diversity in breeding programs because we have a system which is driving fixation of alleles and homozygosity and effectively a reduction in recombination. So managing diversity in breeding programs, looking for rare recombination events, managing haplotype configurations is going to be really important. And coupling that with phenotypic hubs, I think, is a great opportunity. So I think I'll stop there and thank you for your attention. During the week, I've enjoyed the interaction very much, and I'm also enjoying the interaction and challenge of saying, well, how do we fund research and what should we fund? And I think the best way of summarizing that is a quote again from a giant of the 21st century, Sidney Brenner, a Nobel laureate, who summed up the situation with, which type of science to fund is simple. All science is problem-driven and should be judged by the importance of the problem and the quality of the solutions provided. Thank you very much for your attention.